something to watch. So you're going to be looking for these early in the season. From there, they're going to pupate in the leaves, and they're going to either roll the leaves together or they're going to web them together where they can create a protective area where they can pupate. Then the adults will emerge, and when they lay their egg masses, where you'll look for those will be on the surface, the upper surface of the leaves. And from there, you're going to have the development of that summer generation. So if you don't control the spring generation, the summer generation can be really bad. These are the ones that can web those leaves against the berries, causing direct damage to the fruit. And then they'll pupate in the leaves again, and you'll have the adults, which lay eggs for the overwintering larval stage. So here's some examples of tortricid damage to blueberries, and you can see how they'll web things together. On the left side right here, you're going to see how that brown thing in the center is actually a pupa, and in that it's messed in with a bunch of leaves and webbing. In the center, you can see that they can infest the buds, and over here on the right, you can see what they can do to those flowers. You can see the caterpillar right there in the center. One of the main problems with these tortricids is that they are harvest contaminants. So you can see them crawling out of the lugs after you've collected the berries. And so for high infestations, you've got a lot of caterpillars crawling out. In red raspberry, what you notice about damage will be primarily the leaves are going to be webbed together. Look at this photo right here. We'll go back to that in a second. And so oftentimes if you're walking through your red raspberry fields, you're going to see this sort of damage right here. So it's going to be terminal growth often. Those leaves are nice and tender, and when the caterpillars are in there feeding, they can easily roll and web those together. These type of caterpillars are really angry little caterpillars. If you open that up, they'll start wiggling really fast and spin down. And this is what makes them such a problem with harvest contamination. So when you're trying to monitor, you can use pheromone traps to monitor for both OBLR and OT. So in leaf roller management, as far as the eggs and larvae, you can treat them with an IGR, something like Confirm, Esteem, Intrepid. Always check registrations and MRLs of your intended export country. This will affect the larvae that are feeding on those eggshells as well. For the larvae, you've got some other options. So you've got spinosad, and spinosad seems to show similar results to pre-bloom OPs. BT can be really effective. It can be effective, and it really is a nice one to use during bloom time because it doesn't affect the bees. However, you have to be mindful of temperatures with BT. If it's too cool and those caterpillars aren't feeding, then they're not going to get a dose of this. So you may need to make about 3 applications of BT, and you're looking for temperatures above 60 to 65 degrees to get them to feed enough. There are also newer reduced risk insecticides that show good efficacy against larvae, things like Delegate, Altacor. Again, check registrations and MRLs on those. We've been fighting SWD for the past decade. And so they're beginning to see some potential resistance in some of these secondary pests. Spider mites I've seen be showing some problems, as well as leaf rollers could be showing some resistance. So for us, we have put in to have funding in 2020 to do mass rearing of OBLR, and then we'll be able to test for resistance in populations here in Whatcom County in both the red raspberry and blueberry. So there have been resistance developed in OBLR. They're prone to resistance. So you can have pyrethroid resistance, you can have IGR resistance, there could be cross resistance. And so it's something that we'll be looking at hopefully in this next year. So we were able to do a 2019 mating disruption field trial. So what mating disruption is when you put out a pheromone that completely swamps the field with the pheromone odor, and this is going to confuse the males. If that odor of the females is wafting everywhere, then they're not going to be able to locate the females and to mate. And so you should have a reduction in oviposition. So that's the idea behind mating disruption. So there is a product that is already registered in small fruit. It's the OBLR PLR Plus. This is a pheromone twist tie. And so with this one, we put out lures at the rate of around 200 lures per acre. We also put traps, regular pheromone traps in the interior section of that treated field, as well as the outside perimeter area of the field. And we had 2 different controls. So that was the basis of this. It took 4 people about 4 days to put out this amount of lures in 26 acres of an infested area. Let's go back and I want to show you this picture right here. So to apply the twist ties, they have a wire center. There's actually 2 little tubes. One tube has the pheromone in it. If you were to cut it, you could see the liquid inside. So you're going to be twist tying these using gloves. You're going to tie them to the interior portion of the canopy where those moths like to hide. So we put out these, and this is how we evaluated whether mating disruption was effective or not. And so you have to remember, first of all, that mating disruption is a type of a control mechanism, type of an IPM tool that's put on top of something else. It's not the primary tool to manage something. This would be an alternative method that wouldn't add any more residues to the berries. So when you're talking about an insect that could be resistant to some of your insecticides, this is a good approach to take. So the evaluation was pretty tricky. We tried several different things. One of the things we tried was just to look for evidence of the insect in the plants. And so we looked at eggs on the top. We looked at rolled pupae inside the leaves. We looked at feeding damage on the leaves. We also looked at pheromone trap numbers. And we tried putting out virgin moths. To do this, we went out and field collected the pupae. We didn't know whether they were males or females until we looked at them in the lab. We put them in separate little containers. So then when they emerged, we knew those females were virgin females. And so we took those, we put them in little tiny queen cages, put them out in the field at a rate about 10 in the treated area and 5 in the untreated area. And we attempted to dissect these to look at the presence of a spermatophore, but we found this pretty difficult. In fact, we found it impossible. We had moths that died prematurely, even though we supplied them with sugar. We also rode on the harvesters to look for evidence of these caterpillars or any sort of pupal exuviae that might come up on the belt during harvest. And so you can see that it's pretty difficult. Let's look and see the results here. So in this graph, you're going to see the blue is the treatment perimeter, the green is the treatment interior, and then you have this lighter gray or blue color, and that's your untreated area. So straight away, what you're going to see is we put the lures out between 522 and 524. And so by 528, you're going to see that there's already a difference between the interior and the perimeter traps. So remember, those moths are really getting confused on the interior portion because of all that pheromone that's wafting around. But as you go towards the edges, the pheromone volume is going to decrease. That's going to make those moths want to move outward, and any moths that are moving in are going to be captured in those traps. And so you're going to see a difference in the traps on the interior from the perimeter traps. So actually, we achieved trap shutdown, which means that we didn't collect any moths in the interior traps, but we did collect moths in the perimeter traps. There was a spray application about that time, and you can see that the population's pretty well plummeted after that application. Then there was a negligible second flight, perhaps around between the 7th and 20th of August. You know, we were looking for that second generation flight, but we really didn't have any clear indication other than the small flight numbers at that time. So here's a summary of what we found in 2019. We achieved trap shutdown in the interior traps. The moths in the perimeter traps were higher than the interior. We didn't find any worms in the treated field or on the harvester following the lure applications, unlike the UTC. We didn't find a lot in the UTC either, but we did find some. And the second flight was negligible. All right, does that mean that we actually achieved no oviposition? We can't tell for sure. Those virgin moths would have told us that. but this is what we found, and we believe it did have some effect. So here are the pros when you come to OB-LAR mating disruption. It is very compatible with beneficials, a one-time application, so you only have to put those twist dyes out one time. Then you could, you have no resistance problems. You can add additional IPM tactics. And also it's a good management tool for leaf rollers and other crops as well. So we know that historically it's been great in your tree fruit. So the cons would be that right now they're hand applied. It's very costly. It's a supplemental and not a standalone management tool. And you have to remember that OB-LAR is a little bit different than codling moth, which is what they've been using mating disruption on for many years now. OB-LAR is a very strong flyer, and they have a high fecundity. So we're really looking to see whether we had good, strong effects from this this year, and hopefully in the future too. So we do have a next step. The next step is that Pacific Biocontrol has provided us with a prototype puffer, and these are aerosol puffers. They're going to release the pheromone in puffs of about every 15-minute intervals between 5 p.m. and 4 a.m. And for this, you don't have to have experimental use permit as long as it's a straight chain pheromone, and it's up to 250 acres. And so this would be the advantage is that the rate is 1 per acre, not 200. But there are still questions remain. One of them is, puffers have been used in the tree fruit industry, and there's no mechanical harvest there. And so we don't know how the mechanical harvesters are going to act with these puffers, where they're going to rip them off. We plan on securing them to the posts. And so we're going to have to do some preliminary work this next year before we try this puffer technology, just to make sure that there would be no problems with the harvester. But the other thing is perhaps we might have to remove the puffers just before the harvester passes through. And we're looking for an industry commitment for that. One other thing I'd like to mention before I know my time's up. Do they? And so okay. Well, for these, I think putting them on the post would be more secure. Okay. Okay. All right. Yeah, we're gonna. Okay. So we really will secure them, but we'll test it with a harvester at the station as well. One thing I want to mention is a cherry fruit worm. This is another quarantine pest of concern. There are actually 2 fruit worms, but we've not seen damage in small fruit from the cranberry fruit worm. So just the cherry fruit worm. And I wanted to show you this because this is really the typical damage that you'll see if you have fruit worm. You know, fruit worms might be a problem if you have a field that's pretty weedy, but otherwise, if you control your weeds, it might not be that much of a problem. And so you can see one of the main giveaways is this. So you'll see holes, exit holes in the cheek area of the blueberry. And those blueberries that are touching, the worms can actually crawl from one berry inside to the next berry. That's why you have these matching holes. So you can just pull them apart and you can see how they'll match up. They produce a lot of frass inside that berry. You can see it here. And they can also cause premature ripening. So those are some of the signs that you'll be looking for your cherry fruit worm damage. You can also see those tiny eggs. You can see the eggs that are laid on the calyx area. And so when you have the berries, even the green berries out in the field, that's a time when the eggs are going to be laid. One tip you need to know is that instead of using a cherry fruit worm lure, you need to use a false codling moth lure instead. It's more effective. So be sure and use that for monitoring for your cherry fruit worm. And then, how am I doing on time? Okay.
I was quite curious about, I was quite curious about removing them, because if this bait trap gets within the berries, then we got a contamination problem.
That's right. And so those are things we're going to be looking at this summer. There is a pressurized canister in that puffer. And so those are the things that I want to check out this year to see if there's any problem. But, you know, even if you have to jump, you know, stop the harvester just for a couple of minutes while you remove that canister and then put it back, I don't think that's much of a big deal. The harvester's stopping quite often during the field. So we'll see. But if you can add this as an IPM tool, I think it's a terrific one because it's going to cut down on residues that you might be spraying for these leaf rollers otherwise. Okay, I just have a brief amount of time left. We had some issues this year with cutworms. Primarily, they were the spotted cutworm and the winter cutworm. The spotted cutworm adult has a really distinctive wing pattern. You can see those little eyes on either side of the wings. And so if you look at the right over here, you can see that the winter cutworm is an underwing. It has orange underwings, and they're pretty spectacular. You can see those underwings when they're flying. So you can tell those two apart. The immatures have a lot of variability. So when it comes to the Zestia C. nigra, you're not going to have those dark dashes that you have for the, whoops. Let me see. Whoop. Here we go. So you can see the dark dashes right here on Noctua pronuba. So those are the two main cutworms, and I believe that I'm out of time. So thank you very much. And I don't know if we have time for questions.
Okay, now I'd like to go ahead and introduce Camille Holliday. She's the lab director for the experiment station in Mount Vernon, WSU. And she'll be presenting today on caneberry decline study. This is the 3rd year summary of this one.
Good morning. So I'm here to talk about the 3-year study that we did looking at the rate and amount of